Hi, my name is Brighton Mozer.
I'm Peyton van Pevenage.
And I'm Michael Fuller. And our study focused on non-ionizing electromagnetic fields, or EMFs, and the impact they had on an ATR gene expression in Tetrahymena thermophila. So just a brief background of our experiment. First, the model organism we chose was Tetrahymena thermophila. And the reason we did so is it's a unicellular eukaryotic cell that has become a quite popular model organism in biology. It's relatively easy to maintain, and it undergoes rapid cellular division, so that experiments can be performed that may affect its behavior or cellular growth. So non-ionizing EMFs have been shown to disrupt cellular proliferation, and this can potentially lead to cancer. According to one of the studies, when Tetrahymena were exposed to a 60 Hertz magnetic field over the course of 2 days, these Tetrahymena had delayed cellular division and had morphological changes. Now, during DNA replication, single-stranded DNA breaks can occur and prevent the replication fork from progressing. And if this damage is not repaired, it can overall cause damage to the integrity of the chromosome. ATR protein, which we were examining, detects stalled replication forks and initiates a DNA repair pathway. In studies where a conditional deletion of an ATR gene occurred under fibroblasts, this led to the accumulation of DNA double-stranded breaks during the S phase and overall cell cycle arrest. So the ATR protein is extremely important for cell growth to continue to lead to cellular division. Now, the question we were trying to answer was, how does ATR gene expression change in response to suspected extremely low frequency electromagnetic fields induce DNA damage? And how will conjugation behavior, feeding behavior, and overall cell growth change?
Okay, so for the methods, we first made primers for the ATR gene. We used the oligo analyzer software to do this, and these were later used for our PCR reaction. Culturing the cells, they were first grown in an NEFF media, and upon experimentation, they were transferred to an SBP media. Experimental cultures were exposed for 2 rounds to an electromagnetic field with a frequency of 50 Hertz. And this was done for 72 hours for each round. After experimentation, both the control group and the experimental group, RNA was extracted using the Qiagen RNA mini kit. After we extracted the RNA, they were converted to cDNA using the REVERT-8 RT kit, which has a reverse transcriptase. After we collected all the cDNA, we ran a qPCR just to analyze levels of expression for the ATR gene. This was compared to the BTU1 gene expression, which is relatively constant throughout the cell cycle. Cell counting was accomplished using the hemocytometers, and then for the feeding assay, we exposed cells to India ink dye and counted the vacuoles using a hemocytometer.
So for the results, as you can see in figure 1, it's a picture of a microscope slide of our Tetrahymena. And then figure 2 is a diagram of an electromagnetic field. And as you can see in the middle, that's where the electromagnetic field is most uniform. And therefore, that is where we placed our Tetrahymena flasks during the experimental rounds. In figure 3, it is a graph of the average number of food vacuoles per cell that we counted during the feeding assay. And there's control and the 50 Hertz electromagnetic field exposure group. And based on the p-value that we calculated, there was not a significant difference between the amount of feeding vacuoles per cell between the control and our experimental group. And in figure 4, that is a cell growth curve with the number of cells per milliliter of media at 24 hours, 48 hours, and 72 hours for both the control and electromagnetic field treatment group. And based on the p-values, we also found that there was not a statistical significance between the 2 values. But based on our low amount of samples and low amount of replications due to having our experiment cut short with coronavirus, we aren't confident in saying that there is no impact of electromagnetic fields on ATR gene expression. In conclusion, RTq-PCR results showed that the ATR gene was expressed at 2% of the level in the experimental groups when compared to the control group. And the RTq-PCR, feeding assay, and cell counts, again, showed no significant difference between experimental and control groups. Based off our results and the circumstances with low sample size and low replications, we can't say whether or not extremely low frequency electromagnetic fields increased or decreased the transcription rates of the ATR gene. And for future directions, we would recommend considering running more RTq-PCR rounds. And we also think that it would be interesting to explore the effects of a change in frequency on the genes, whether that be lowering the frequency of the magnetic field or increasing it. We would like to thank Dr. Ottohit for her guidance during this whole process, and for Dr. Spilka of the physics department for allowing us to use equipment to run our experiment.